So students, today in this lecture, we start a topic microsporogenesis from our class 12th chapter number 2nd, sexual reproduction in flowering plant. So students, let's start the topic microsporogenesis. So firstly, we define the term microsporogenesis. So simply, microsporogenesis is a process of formation of microspore from microspore mother cell through meiosis. The process of formation of microspore from microspore mother cell through meiosis. Microspore sporogenesis is a process of formation of microspore. The ploidy of this microspore is haploid and the microspore mother is diploid. In simple words, we represent that this is the microspore mother cell, or we can say that pollen mother cell. Okay, so by the process of meiosis, the pollen mother cell produce microspore tetrad, the haploid microspore tetrads. These microspores initially are attached through callus. Initially attached through callus. When the anther is dehydrated or dehiscence performed, the callus is digested by Callus enzyme. This enzyme is secreted by tapetum. Secreted by tapetum layer, which digests callus. By this we get, when the anther dehiscence is occur, we get free microspore, free haploid microspores. Okay, these are haploid microspore, free haploid microspore. So, formation of microspore from pollen mother cell through meiosis process is known as microsporogenesis. So, next we start the microgametogenesis. In simple words, this process is known as microsporogenesis, formation of free haploid microspores from pollen mother cells through meiosis. So students, let's we discuss about how the pollen grains are formed, or we see that how the male gamete in flower is formed. Next is microgametogenesis. Means how the formation of pollen grains, how the pollen grains are formed. So through this process, we see that formation of pollen grains or male gamete. These pollen grains are the male gametes. Okay. So So let's discuss the process of formation of pollen grains. We take one haploid microspore. One haploid microspore. Okay. So now the haploid microspore perform mitosis. Okay, here the mitosis processes occur for the formation of pollen grain or male gametophyte, yeah, male gamete. So now, by this, there is a formation of male gamete. Here, one is vegetative or one is germinative cell. Vegetative and one is germinative cell are formed. As we all know that by one mitosis process, we obtain two cells. Okay, or by one meiosis, we obtain four cells. Clear? So, we get one vegetative cell and one is germinative cell. Here, the vegetative cell are larger in size. And the germinative cell is smaller in size. Okay. So, this is two-celled pollen grains. This is two-celled pollen grain. In 60% of angiosperm, two-celled pollen grains are present. And in 40% angiosperm, there is a tricellular pollen grains are present. So, here this for tricellular, here the second mitosis division is occur. The pollen grain is the germinative cells are divided before the pollen grain are sheathed on the stigma. So, here the vegetative cell remains same, but the germinative cell is divided into two cells. So, it is a tricellular form. Clear? So, for the formation of a pollen grain from a microspore, we required two mitosis division. To getting the tricellular form of pollen grain. Clear? So now we discuss about this pollen grain. Here the pollen grain is formed. Two-shelled pollen grain. Okay. One is inner lining and one is outer layer of the pollen grain. Clear? This is your exine, outer layer, or this is your intine, inner layer of the pollen grain. This is outer layer and this is inner layer. Okay, so the exine is discontinuous. Here a space which is called germ pore, through which the pollen tube is formed. So here is a germ pore where exine is absent. Here exine is absent. So students, we say that exine is discontinuous and intine is continuous because there is no area where intine is absent in the pollen grain structure. So exine is discontinuous layer and intine is continuous layer. Okay. Second thing is that the exine is made up of sporopollen. The exine is made up of sporopollen. This sporopollen substance is highly resistant in our environment. Resistant substance. Due to presence of this sporopollen, the pollen grains are stored. Okay. Second one is intine is made up of packaging and cellulose. Made up of packaging and cellulose. Okay. So these are the two layers of the pollen grains. One is intine and one is exine. Exine is made up of sporopollen, which is resistant substance, and intine is the inner layer, which is continuous layer made up of pectin and cellulose. Clear? So this structure is, we call that pollen grain or male gametophyte. Male gamete, male gamete, sorry. Okay. And the last thing is that the size of pollen grain is approximately 25 to 50 micrometer in diameter. So this is simply about the formation of pollen grains, how the pollen grains are formed. Okay, so thank you so much.